Hello everyone, and thank you for joining our webinar titled The Growth of Supplement Trends presented by Neotron. My name is Massimo Veronesi. I'm the head of GC and LC Nutrition Department and product specialist for supplement. Today I'll be your host. With me there is Eva Topciu, the team leader deputy for the sales, and today we'll cover valuable insight into latest trend in supplement testing and quality control. Before starting, a very brief introduction to our company. Cotecna is a Swiss company located in Geneva, is a leading provider of testing, inspection, and certification. Cotecna has more than 7,000 employees and more than 108 offices across the globe. You can say that the entire team offers expertise in 5 main areas: agriculture, food safety, metal and minerals, consumer product, and certification. Neutron is part of Cotecna since 2019, and it is the biggest lab of the group. We are more than 550 employees that work in 10,000 square meters of lab. We handle more than 1,000 samples per day, and we perform more than 800 methods that involve contaminants, both chemicals and microbiological, additives, nutritional labels, and packaging. There are the field of testing activities which Neutron has activated throughout its history in relation to the market and obviously to the customer request. Well, now I leave the floor to Eva. If you have any question during the meeting, feel free to use our Q&A section and the chat box. So please, Eva, thank you.
Thank you, Massimo. Hello, everyone. Thank you so much for joining us today. It's a pleasure to speak with you about a topic that had seen such a remarkable transformation in recent years, namely the global rise in dietary supplement consumption. Over the past decade, the global supplement industry has expanded rapidly, evolving into a multi-billion-dollar industry that shows no signs of slowing down. The interest in supplements is no longer limited to health enthusiasts. It has become a part of everyday wellness for many people around the world. Market data show that North America remains the leading region, followed by strong growth in Europe and the Asia Pacific. Meanwhile, regions like Latin America, Middle East, and Africa are still small players, but the demand there is picking up fast, and there's a lot of potential for future growth. Yet, with this rapid growth comes a new set of challenges. For supplement producers, there are several key considerations to ensure in order for their products to meet necessary standards such as ingredient verification and quality and safety standards. And to do that, they rely heavily on trusted, experienced labs. These laboratories play a key role in testing and confirming that everything checks out, from what's on the label to what's actually in the bottle. Together, we will examine first the key active ingredients that are commonly used in dietary supplements. Second, the critical compliance tests that help verify what's stated on the label is actually accurate. And finally, the safety evaluations that check for any contaminants or hidden substances. The categories that we will go through are, first of all, immune support supplements. Then we've got cognitive enhancers. We will move on to sleep supplements. Next up, longevity and cellular health, joint health, weight management, and finally, sports performance supplements. So let's begin with immune support supplements. These supplements are meant to help strengthen and balance the immune system. They usually include a mix of vitamins, minerals, herbs, antioxidants, and other active ingredients that work together to support overall immune health, such as vitamin D3, vitamin K2, quercetin, and elderberry. It's important to mention that high quality formulations require a wide range of quality checks. Now, let's take a closer look at examples of our analytical approach to testing the supplements and walk through some of the most commonly used techniques for compliance testing. One of the main techniques we rely on for the determination of vitamins is high performance liquid chromatography coupled with a UV detector. This method is precise and reliable, which makes it ideal for quantifying a wide range of vitamins in complex matrices. Now, a key part of this technique is the UV spectrum, which helps us understand how a compound absorbs light at different wavelengths. Each compound has a unique UV absorption profile, almost like a fingerprint. So, by selecting optimal or multiple wavelengths, we enhance sensitivity and detect even minor impurities. As a result, this improves the accuracy and the robustness of both identity and purity testing. The same analytical approach is applied also for identifying and quantifying quercetin in complex plant extracts. So, by comparing UV profiles and retention times to certified reference standards, this method really allows us to distinguish quercetin from other similar flavonoids, ensuring the authenticity and the quality of plant-based ingredients in supplements. So, moving on to analyzing botanical extracts like elderberry, a combination of analytical techniques is essential for achieving both efficiency and precision. Typically, the process begins with identifying key biomarkers, such as anthocyanins. These polyphenols determine the extract's quality, authenticity, and potency. Now let's take a look at our methods. The Follin-Chocalteau assay is a spectrophotometric method that is useful for rapid, cost-effective screening of total polyphenol content. This type of method is ideal for monitoring batch-to-batch consistency and providing a general overview of the extract's quality. However, this method is non-specific and also detects other polyphenols such as catechins, flavonols, or tannins. Now, in order to perform precise identification and quantification of individual anthocyanins, we need to employ chromatography. HPLC-UV is our method of choice to detect anthocyanins with high resolution and sensitivity. That said, there's a limitation. It relies on authentic reference standards, which may not be available for all anthocyanins in complex extracts. So, as a result, some peaks detected by HPLC-UV may remain unidentified. Also, we have to consider another aspect that elderberry is such a complex extract, and the detection in HPLC-UV may be prone to interferences. To address this issue, what we do is employ liquid chromatography coupled with mass spectrometry. This technique combines the separation capabilities of HPLC with the sensitivity and specificity of a mass spectrometer. LCMS can identify anthocyanins with great certainty by providing both molecular weight and structural information, which means it allows us to detect compounds that HPLC UV might miss or misidentify. Despite its strength, LCMS also faces challenges with the standard availability. While it provides better structural details, the identification of anthocyanins is limited due to the fact that we don't have reference standards available, especially for anthocyanins that are new or not well studied. But actually, the true effectiveness of this analytical strategy lies in its layered approach. Spectrophotometry serves as a rapid screening tool, while HPLC-UV and LCMS deliver targeted, confirmatory analysis. As a result, this synergy creates an efficient workflow, and the cross validation between techniques enhances the confidence in the results and the characterization of botanical extracts such as elderberry. Now, since we're a laboratory that thoroughly tests dietary supplements, we understand that ensuring their safety is absolutely essential because these products are directly consumed by individuals. But then the question becomes, how can a manufacturer be certain that the products they offer are safe for consumers? Well, the answer lies in comprehensive contaminant testing. One specific health concern in dietary supplements is heavy metal contamination. It is well known that decades of industrial activity and pollution have introduced heavy metals into the environment. They raise great concern because they can be absorbed by plants and as a result may enter the human diet through food or plant-based supplements. And since most supplements include plants and botanical extracts as active ingredients, it is vital to verify that they are free from harmful levels of heavy metals in order to ensure compliance with regulatory standards. These contaminants are often present at very low concentrations, so it is crucial to select a sensitive and reliable analytical technique. For this purpose, our method of choice is inductively coupled plasma mass spectrometry. Typically, the samples are digested under highly acidic conditions and then introduced into the instrument, which measures the elemental mass of heavy metals with high precision. Therefore, we can reach a very low limit of quantification that can detect trace levels of heavy metals. Now, let's move on to another category, namely cognitive enhancers. These are the supplements that are designed to improve memory, focus, and overall mental health. Some of the most commonly used active ingredients are bacopa, citicoline, and phosphatidylserine. The quality of ingredients is crucial in order to deliver these benefits. Therefore, it is essential to ensure that each component is sourced, standardized, and tested for purity and safety. For instance, Bacopa Monieri is commonly standardized to 20-55% of bacasides that are the active compounds. They require precise quantification to ensure consistent potency and correct dosing. Usually, the trusted method for this purpose is the official USP assay. USP assay, which uses HPLC UV to separate and measure total bacasides. Meanwhile, the quantification is based on USP certified reference standards. This approach ensures product consistency, potency, and compliance. This test is highly valuable due to the fact that USP certification is widely recognized across many countries and supports both international trust and product acceptance. Now, a quick mention to the analytical approach for detecting phosphatidylserine. The determination of this active ingredient is effectively performed by using HPLC coupled with evaporative light scattering detection. In particular, this technique is well suited for compounds that lack strong UV absorbance, such as lipids and phospholipids. Now, let's look at the process. The sample is separated by HPLC, the mobile phase is evaporated, and the resulting light scattering from the analytes is measured to determine the compound's concentration. The advantage of ELSD is that it offers high sensitivity in detecting lipophilic compounds without relying on UV absorbance. Now, let's shift our focus to the safety aspect, which is crucial since plant-based ingredients are employed in various formulations. One of the major contaminants we need to watch for in botanical extracts are mycotoxins, especially aflatoxins and ochratoxins. These harmful compounds are produced by molds that can grow on plants at any stage, from cultivation to storage. And since they pose serious health risks, they are also strictly regulated. Therefore, it is crucial to detect them accurately and quantify these contaminants to ensure both the safety of raw botanical materials and compliance with the standards. For this purpose, we rely on advanced methods such as LCMS. LCMS can detect and quantify mycotoxins in complex botanical extracts with high precision. While also other techniques may be effective, but LCMS stands out due to its unparalleled sensitivity and ability to detect a broad spectrum of mycotoxins in a single acquisition. It identifies and quantifies with high accuracy at trace levels. By using these advanced techniques, we can ensure botanical extracts are free from any harmful level of aflatoxins and ochratoxins and demonstrate the product safety. Now, let's move on to another category that is rising in popularity, which are the sleep supplements, or also known as sleep aids. Okay, so when it comes to sleep supplements, there are several types of ingredients on the market. However, modern formulations often focus on three key ones, which are melatonin, magnesium in the forms of magnesium glycinate and L-threonate, and compounds that derive from specific herbal extracts, such as L-theanine from green tea. The reason these ingredients are preferred is because they naturally support the body's sleep-wake cycle without acting as strong sedatives. Therefore, the dosage and the quality of the ingredients are key aspects for this type of formulation. And it's crucial to verify label claims and potency. So, to assess magnesium content in supplements, our method of choice is inductively coupled plasma optical emission spectrometry. Next slide, please. So this technique works by nebulizing the sample in a high temperature plasma and then measuring the light emitted by magnesium atoms at characteristic wavelengths. This allows for precise quantification of the total elemental magnesium present in the sample. There are several advantages to using this technique for magnesium testing. It can detect magnesium content with high precision. It handles complex matrices well. It is also suitable for both finished products and raw materials. So, another big advantage, of course, is also the rapid processing of many samples. So to sum up, ICP OES provides a powerful, fast, and reliable way to quantify magnesium. On the other hand, to verify melatonin dosages, next slide, please. To verify melatonin dosages in supplements and the purity of raw materials, the best technique we rely on is HPLC coupled with a fluorescence detector. This technique combines the HPLC separating capabilities with the selectivity of a fluorescence detector, which identifies melatonin by exploiting its natural fluorescence when exposed to certain wavelengths of light. This is actually a significant advantage because not all compounds exhibit fluorescence. Only a few, like melatonin, can absorb light at one wavelength and emit it at another. This unique property provides with inherent selectivity and sensitivity. Therefore, we can ignore non-fluorescent impurities or matrix components that might otherwise interfere with accurate detection. Now, let's go through the safety aspect of dietary supplements or botanical extracts. One crucial factor to consider for product safety is the presence of residual solvents, since they are thoroughly used during the manufacturing process, such as extraction, purification, or formulation. Even though solvents are essential for producing many ingredients, they must be carefully controlled because some of them can be toxic even at low levels. The standard method we rely on for accurate detection and quantification is gas chromatography with flame ionization detection. Since almost all organic compounds ionize well in a flame, GCFID is a highly sensitive technique to detect hydrocarbons and organic solvents. The sample is injected in the gas chromatographer, vaporized and carried by an inert gas through a chromatographic column where the different solvents are separated. Afterwards, they enter the flame ionization detector, which combusts the compounds and measures the resulting ions. So, by adjusting some chromatography parameters, for example, the column temperature, the detector, and so on, we are able to identify and quantify a wide range of solvents in a single acquisition. On your screen, you can see a list of some of the compounds that we detect divided on a classification of solvents by their toxicity risk. Now, let's move on to another category, which is quite popular, the longevity and cellular health supplements. These formulations are designed to delay or reduce age-related decline at cellular level. While these supplements may contain a variety of ingredients, the most notable active compounds are resveratrol, astaxanthin, and spermidine, which stand out for their distinct benefits. Now, let's dive deeper into supplements containing astaxanthin. For these supplements, is essential to perform rigorous quality and safety assessments. The first step is identity verification. Natural astaxanthin is primarily sourced from Hematococcus pluvialis and is mostly present in mono and diesterified forms. High performance liquid chromatography coupled with UV visible detection is our method of choice for this purpose. The technique relies on its distinct absorbance maximum and characteristic spectral profile to confirm the identity of astaxanthin. Importantly, HPLC separation is capable of distinguishing free astaxanthin from its esterified derivatives based on difference in polarity and retention times. While this technique can resolve and quantify esterified and non-esterified forms, it does not provide direct identification of the specific fatty acids involved without additional mass spectrometric analysis. Nevertheless, it provides valuable information to differentiate natural esterified astaxanthin from synthetic astaxanthin, which is not esterified, and by this we support quality claims. Safety-wise, for supplements made with natural ingredients like astaxanthin, microbial testing is one of the most essential steps in quality control. Plant and algae-based ingredients are naturally prone to microbial contamination at various stages, from cultivation to storage, and they can pose serious health risks, including allergic reactions or toxicity. Moreover, microbial contamination doesn't just threaten consumer safety. It can compromise the stability and potency of the active ingredients as well. For example, astaxanthin is sensitive to oxidative degradation, and microbial metabolism can accelerate the breakdown of this carotenoid and reduce the compound's antioxidant capacity. Many critical tests are routinely performed to assess microbial safety, such as total aerobic count, yeast and mold count, and pathogen detection. We rely on plate-based methods and PCR-based methods. Particularly, PCR can detect specific DNA sequences of pathogens and offers accurate results within hours compared to days for culture. It can even detect viable but not culturable organisms. And to perform these tests, we can rely on ISO methods or official pharmacopoeia methods. Each of these tests help ensure that raw materials and final products meet both quality standards and regulatory requirements. Now, let's move on to the next category of supplements, which is joint health supplements. These formulations often combine several active ingredients that work synergistically to improve joint function. Among the most and effective ingredients, we have under nature type 2 collagen, curcumin, hyaluronic acid, MSM, glucosamine, and chondroitin. These ingredients require a robust set of tests to ensure quality, safety, and label accuracy. Since they each have different origins, like animal, microbial fermentation, mineral, botanical. So, for instance, under nature, type 2 collagen in supplements is verified through HPLC-UV. The sample is subjected to controlled denaturation into amino acids, followed by derivatization and quantification using this technique. This method allows us to accurately determine hydroxyproline levels, and based on EU regulatory definition, we then convert the hydroxyproline result to determine the total collagen content. Meanwhile, hyaluronic acid and glucosamine are analyzed using HPLC with pulsed amperometric detection. This technique is highly sensitive for detecting carbohydrates and amino sugars with precision, even at low concentrations. For MSM, we use gas chromatography with flame ionization detection to identify and quantify these compound. This technique provides robust sensitivity for small volatile molecules like MSM. Chondroitin sulfate is usually analyzed using HPLC with UV detection, which helps identify and measure these compounds accurately. Chondroitin, it can come from different animal sources like bovine, porcine, avian, or marine, each with slightly different structures. What we do is we compare the sample's chondroitin peak profile to the chondroitin present in the reference standards that derive from the same source as our sample. This approach ensures the supplement is authentic, properly labeled, and meets both identity and potency requirements. Moving on for turmeric extracts used in dietary supplements is essential to confirm both the identity and the standardization of curcuminoids, which are the primary bioactive compounds, and they are typically standardized to 95%. High performance liquid chromatography is our method of choice since it efficiently separates and quantifies the three main components, namely curcumin, dimethoxycurcumin, and bis-dimethoxycurcumin. This analytical approach provides a precise profile of curcuminoid content for consistency with label claims. In addition to verifying active compounds like curcuminoids, contaminant testing is a critical step in ensuring overall safety for turmeric-based supplements. Among the many tests that we perform, pesticide residue analysis is especially important for such extracts. Turmeric, sourced from conventional agriculture, is particularly vulnerable due to intensive farming practices. To address this, we offer comprehensive multi-residue pesticide screening for which is capable for detecting and quantifying hundreds of potential contaminants in a single acquisition. Our testing employs advanced techniques such as gas chromatography and liquid chromatography, coupled with tandem mass spectrometry. These techniques deliver high sensitivity and specificity even at trace levels measured in parts per billion. These tests are essential for detecting residues and ensuring adherence to global maximum residue limits. We select our compounds based on targeted risk assessments for each botanical species, along with international alerts and regulatory priorities. Our analyses follow residue definitions established in the EU regulations. For example, the EU expresses malathion content in botanical species as a sum of malathion and maloxon, which is its metabolite, and we determine both accordingly. We also follow pharmacopoeial guidelines, including those from USP and European Pharmacopoeia, to verify compliance for botanical raw materials like turmeric. Usually, these methods require screening for hundreds of pesticides at very low thresholds. And these are on the screen. Here are some of the major pesticide groups typically included in these pharmacopoeia standards. Now, let's shift our focus to another category, namely weight management supplements. These formulations have become increasingly popular as more people seek effective solutions for achieving and maintaining a healthy weight. Key ingredients like berberine, epigallocatechin and capsaicin have gained attention for their proven benefits. For identity and potency tests on supplements, we conduct the analysis by HPLC-UV to confirm the presence and quantify these three compounds. So, regarding berberine and capsaicin, we use HPLC-UV that separates the compounds based on their unique chemical structure and measures the absorbance at specific wavelengths to determine the concentration. Meanwhile, epigallocatechin also is typically quantified using HPLC UV detection to ensure both raw materials, like, for example, green tea extracts, and finished products contain the right levels of this key catechin. Unlike basic spectrophotometric methods, HPLC separates epigallocatechin gallate from other similar catechins like apicatechin or apicatechin gallate and so on, and offers much higher accuracy and specificity for verifying ingredient content. Now, let's move on to the safety aspect. Since these ingredients are plant-derived, there's a risk of contamination with ethylene oxide, which is a chemical often used in fumigation and sterilization process. It is classified as carcinogen and mutagen by major health authorities. This is why it is strictly regulated and its maximum residual limits have been set for food and supplements. Therefore, to ensure safety and compliance, we use gas chromatography with mass spectrometry. With this technique, we can detect both ethylene oxide and its byproduct, 2-chloroethanol, which is a key marker of its usage. Now, let's go through the process. This method vaporizes the sample, then it separates it in a gas chromatograph, and finally uses a triple-quadruple mass spectrometer to identify and quantify these compounds with high precision, even at levels well below the regulatory limits. And finally, we have gone through the last category left to explore, which is sports performance supplements. Now, these products are designed to optimize physical performance, enhance recovery, and support overall athletic goals. Among the most popular ingredients in this category, we find creatine monohydrate, citrulline malate, beta-alanine, and palmitoyl ethanolamide. For these supplements, accurate dosing is essential to prove their effectiveness, but also their safety. This makes quality testing a key step in verifying claims and ensuring products are free from contaminants. Take creatine monohydrate, for instance. It can be affected by impurities during manufacturing or improper storage. One notable impurity is creatinine, which is a degradation byproduct formed when creatine is exposed to moisture, heat, or acidic conditions. Elevated creatinine levels may indicate poor manufacturing or storage practices and can signal reduced product potency. To accurately differentiate between creatine and creatinine, our method of choice is high-performance liquid chromatography with ultraviolet detection. This method exploits the distinct chemical structures of the two compounds to separate them and their characteristic absorption spectrum to identify them. Currently, there are no specific regulatory limits for creatinine presence in creatine supplements. However, as I said before, it can be an indicator for improper manufacturing or storage. So, if the raw material has showed a low potency, it may be necessary to conduct further creatinine testing. High-quality creatine monohydrate raw material typically contains minimal impurities, so testing for creatinine can serve as an important quality control step. Now, another test that we routinely conduct is gluten testing, particularly for these products which are often produced as powders or ready-to-drink formulas. Gluten contamination is a major concern, especially for products that carry gluten-free claims. Moreover, cross-contact during manufacturing or the use of ingredients derived from wheat, barley, or rye can introduce gluten into the final product. So it's important to mention that in regulated markets like the US and the European Union, supplements labeled as gluten-free must contain less than 20 parts per million of gluten to be compliant. On the other hand, for those that do not make such claims but may contain gluten, proper labeling is required because we need to alert consumers of potential contamination. To ensure accurate detection, we employ enzyme-linked immunosorbent assay, also called ELISA, which is a highly sensitive and specific technique capable of detecting gluten levels well lower than the declared limits. ELISA allows us to differentiate between gluten sources, ensuring a precise assessment of potential contamination. Now, let's reflect on what we've explored today. We are a laboratory that supports the supplement industry, and we understand that innovation in formulation must be matched by innovation in testing. From identity and potency to contaminants and allergens, for us, quality verification is not just a regulatory checkbox. It's actually a foundation for consumer trust and brand success. We have over a decade of experience in supplement testing, and we have supported brands through every stage of development. Beyond testing, we provide expert consultation to evaluate results and really guide the next steps. In the current world, the market is evolving rapidly, and so are we. We are staying ahead by investing in cutting-edge technologies, expanding our capabilities, and we stay aligned with emerging regulatory updates. Therefore, our mission is to be more than just a lab. We are your quality assurance partner, and we're committed to shaping a safer, smarter future for dietary supplements. Thank you. Thank you for your time and your attention. I'm happy to pick up any questions that you have.
Thank you very much, Eva. It was a very interesting presentation. So during your webinar, we received some questions, so we can start from the first one. Do you perform tests to determine the bioavailability of active ingredients in supplements?
So we don't currently perform direct bioavailability studies. Usually, These type of methods are carried out in clinical and specialized research settings. However, what we do offer is dissolution testing. This kind of testing evaluates how an active ingredient is released from a supplement under simulated physiological conditions. So, even if it doesn't measure absorption in the body, it can be a key indicator of how readily a compound becomes available for absorption. And it's an essential step in assessing formulation performance and consistency.
Thank you. The next one. What's the best way to determine the shelf life or expiry date of a supplement?
Okay, the best way to determine the shelf life or expiry date of a supplement. So, the most reliable way to determine the shelf life is through real-time stability studies or accelerated stability studies. These type of tests evaluate how the product holds up over time and under specific environmental conditions, typically carried out with variations in temperature and humidity. At our lab, we can design customized stability protocols to monitor key parameters, for example, the potency of active ingredients, the physical changes, and, overall product integrity at different time points. And based on the results that we obtain, we can estimate how long the product will maintain its quality and efficacy, which is essential for setting an accurate expiry date.
Thank you. The third one. Are there any raw materials that present particular challenges for MOSH/MOA analysis? Which ingredients can influence the MOSH/MOA measurement results?
Okay, MOSH/MOA. So, the most challenging matrices are for sure plant extracts, but also natural oils and waxes and antioxidants. So, all of these ingredients can give an overestimation of the final results due to the presence of natural interferences that can be removed with the cleanup procedures that are used for the moshmo extraction. And moreover, it's not always possible to standardize these methods because these samples, these products have a wide variability and complexity because they contain a multitude of natural compounds with different analytical behaviors. So, to overcome this, we employ two-dimensional GC technique. The GC by GC TOF mass FID technique allows both spectral characterization of mineral oil markers, which are well distinguished by all natural compounds, and also their quantification, respectively, provided by the combination of TOF-MS technique with FID detection. So,
Thank you.
if you're interested in the MOSH/MOA topic, my colleagues that are expert in this analysis covered it in a previous webinar, and you can find the registration on our YouTube channel.
Thank you. And what is the recommending technique for identifying botanical species present in powdered turmeric?
Identifying the botanical species. Okay. Well, there are several methods available for botanical species identification, especially for turmeric. But one of the most effective techniques that we employ is NGS, next generation sequences, which uses a method called metabarcoding. This approach analyzes DNA fragments from the sample and compares them to reference databases to identify the different plants species that are involved in the sample. It is particularly useful when multiple botanical ingredients are present because it provides a comprehensive and reliable picture of the product's botanical composition. And for those who would like to dive deeper into the authenticity topic, my colleagues are also presenting the next webinar that will cover this authenticity topic in a much greater detail.
Thank you very much, Eva. Thank you very much. And well, as we near the conclusion of our program, I hope that you found this webinar useful and interesting, obviously. I really want to thank you for your attention. Thank Eva for her effort. And in the next days, we will send you a survey, obviously a recording of this webinar and the relative presentation. So thank you again and have a great day and see you
Thank you.
in our next webinar.